Good evening, everybody. So today, the talks have been very interesting so far. We're all looking at what to do in this pandemic of COVID-19. IGeneX, who specializes in Lyme testing, decided to help out and go into COVID-19 testing using what we have learned from Lyme over the last 25 years. And today I'm going to give you an overview of what IGeneX is doing to help out. So basically, I think you already heard about the SARS-CoV-2 genome. It's about 30,000 base pair. The three major genes are S gene, the outer surface proteins, and the nucleocapsid. Those are the two that are very important, and the ACE protein. And as you have heard, there is two ways of looking at any disease. One is to look at what causes the disease, and another one is to look at what the host response is, just like any other diseases. So in case of COVID-19, of course, what we are going to look at is RNA and not DNA, because it's an RNA virus. So for direct testing, one can culture, but that's not something that's practical. The quickest way of looking at it is either to look for antigen detection or look at RNA or DNA by amplification. And I will be talking about looking at RNA by amplification as a direct test. In terms of indirect test, we've heard about serology quite a bit today, about RDTs, ELISAs. Those are the two. What I am going to cover for indirect tests, what we are working on is immunoblots. So basically, I'm going to first start out with a PCR test, which I believe is something that one has to do. It's very quick. One gets an answer on the same day. With what we know now, one should really take a panel approach. And I think that was pretty clear from everybody's talk that one test is not enough to answer questions for every patient. Every patient can be at a different stage of the disease, could be a carrier, could have had the disease. So what do we do? Early in the disease, viruses are going to be present. And in this case, it's a respiratory virus. So we are looking at nasopharyngeal swabs early in the disease, or maybe a bronchial Levat sample has been suggested, aspirates have been suggested. So different things can be used, but the most common one currently being used is nasopharyngeal swab. Early in the disease, we have the virus, then the T cells are present. So when an infection occurs, the first response is T cells. And one can look at the T cells that are activated by the virus. Currently, there are no tests that's looking at T cells, but I am sure there are several companies developing those tests. So what do we have as an early marker? PCR is the choice of test, RT-PCR. As was mentioned earlier, IgM's antibodies start being produced fairly early in the disease, about 5 days, and they are present for a while. Then you get an IgG response. So in order to look at the full spectrum of the disease, one should really consider doing a panel approach, especially with what we know today. There are carriers around us. There are people who have recovered from the disease, people who might just have got an infection. So that is what the approach IGeneX has taken. Our approach is to look at, and we have developed two sets of tests are being developed. One is PCR, which is ready, and we are currently working on immunoassays. So for PCR, what are we using? We are using COVID-19 real-time RT-PCR. And we are using, this test targets three proteins, three RNA markers, S protein and protein and ACE proteins. I'm not going to go into details about it. I think we already talked about that. The specimen type currently that we have tested and can use is nasopharyngeal swab, oropharyngeal swab, or any sample that's collected in CDC or FDA approved collection kits. Shipping, we require it to be shipped cold. We are looking at transport medium that does not require any cooling, and the sample is very stable for a much longer time. The preliminary data is very good. If that works, that's the collection medium we will be shipping out to our clients. And we guarantee the turnaround time would be 24 to 48 hours mostly once we receive the sample. Positive results would be reported out immediately, and this test will be considered stat. Negative results will be reported later. Because I think getting the physician and the patient knowing that they have the infection is very important in this case, especially if it's a PCR test, we are looking at active disease. So what we do is we extract total RNA from the patient sample, the swab, and then we amplify the purified RNA by using multiplex real-time RT-PCR. The test from beginning to end takes 3.5 hours. The limit of detection is 12.5 copies per PCR of purified RNA. This translates to about 100 copies by PCR in a clinical sample. Per mil of sample, 5,000 copies we can easily see by our method. We've done a clinical study on 47 samples to date. 3 of them were positive for COVID-19. We tested influenza A, B, and RSV. All the samples positive for these other flus were negative, and we looked at 17 negative samples. They were negative. The specificity is 100% based on this small study. Sensitivity was 100%. Again, 3 is not a big number. We do need bigger size to sort of comment on the sensitivity. The 2 test codes we use are C100, C200, and the price is $135 per test. Indirect testing. ELISA, IgM, and IgG, we are working on that. Immunoblots, IgM and IgG, we are looking at several COVID antigens on this, COVID-19 antigens. We hope that this test will be ready soon. Why have we gone for immunoblots? The reason is we have a lot of experience with immunoblots. ELISAs are good for screening. The problem with ELISA we find is the sensitivity and specificity. Immunoblots have far superior sensitivity and specificity, so we are using our expertise in developing immunoblots for COVID-19 testing. We expect that the test will be ready and validated by middle of April. And as I mentioned earlier, why the panel approach? Because we don't know at what state the patient is. Therefore, in order to find out whether the patient has active infection, was exposed, or is a carrier, we recommend a panel approach. Currently, we offer only real-time PCR test. By the middle of April, we should be able to offer ELISA and immunoblots. But my recommendation, based on what I've always seen over the last 25 years, is to do PCR with immunoblots would be the right way to go around. And we should be able to turn around the results for PCR, as I said, between 24 to 48 hours in most cases. The positive results will be reported stat. As soon as we have the result, we are going to inform the physician about it. Immunoblots probably will take about 2 days to do. So these tests are going to be very rapid. Results will be reported very quickly. And I hope that this helps the patients out there and the physicians. You know, this should be a good set of tools to look at COVID infection in patients, and also for epidemiological studies, this could be very useful. So if our contact information is right here. If you have questions, you can email me or call us. And with that, I will end. If anybody has any questions, please ask. Thank you.
The question on most of the labs that have been discussed tonight using a proprietary lab that has patient rights for lateral flow, meaning are they getting testing serology that we can get directly into our office through McKesson, Henry Schein, etc.?
This is not, we are not, at this point, we are not looking at lateral flow assays that can be sent to doctor's office. This is something that we would be doing in our lab. Immunoblots, unfortunately, cannot be done in a doctor's office. It's not that simple, but you know, if somebody's sending a PCR sample, it's easy to send a blood sample and a swab at the same time and get the answers very quickly.
Another question, do you have a price for the immunoblot test?
The price would be around $175 to $200 if you did it separately for immunoblots, and $135 for the PCR, but we are looking at bringing the price down. I don't have the numbers. If you do a combo test, it should be, you know, because I really believe that panel approach is the right way. So we are trying to figure out how best we can, how low pricing we can bring this to. It could be anywhere between $250 to $275 for the whole panel, PCR and 2 immunoblots.